Dr. Lee is a retinal specialist at the Ottawa Hospital and an assistant professor at the University of Ottawa. He completed his ophthalmology residency at the University of Alberta and his medical and surgical retinal fellowship training, at the prestigious Wilmer Eye Institute at Johns Hopkins University in Baltimore, Maryland. So please welcome Dr. Tom Lee.
Thank you very much. When I was first asked to do this presentation, I was kind of surprised because I'm not trained in genetics, and but as a retina specialist, I see many genetic patients. Personally, my wife's mother, or my mother-in-law, she has retinitis pigmentosa. She has RP. And interesting enough, my wife's godmother also has RP. And right now, I'm still not sure if my wife herself has RP or not. So it is a subject that is very personal to me. Before I begin with all the nitty-gritty, I'm going to talk a little bit or give a description as to what one of my patients told me. This is a 29-year-old lady out of art school, very enthusiastic about life, and she has retinitis pigmentosa. And she came and saw me last month, and she was quite frustrated and felt a little bit down. And she told me, I can't do my illustration work anymore. I can't draw straight lines. I can't make the lines meet up. Things look distorted to me. I'm no longer able to take on clients and take on new contracts. I can't see print very well. When I look at words, I only see a couple of letters at a time, and I try to remember those couple of letters and before seeing the next few letters, and then try to remember to put them back together to make up the word. I can't read music anymore, and I can't keep up with the songs in my choir group. I can't visually recognize the food on my plate and have other people verify what I'm eating. I often bump into objects on my right side. My visual field has decreased so much that I constantly bump into my own furniture at home. If objects are placed just 3 feet in front of me, I will still bump into them, and I can't see well enough to avoid them. Even in good light, I have trouble seeing the details on my palm, and I can barely see the tips of my fingers. When I look into the mirror, my face is just a smudge. The glasses that were prescribed to me, and I use amber glasses, they tend to help with decreasing the light sensitivity, but now I'm finding them not working very well anymore. So this is a description from a very young lady who has a full life ahead of her, and she can't see very well, and she can't see well enough to work, and it's affecting every part of her life. So that's a little bit of an introduction to this talk, and as to why research in the genetics of eye disease is so important. To change gears a little bit, I'm going to continue to describe why we have genetic eye disease, and why should we start testing for genetic eye disease, and should we do therapy on these patients. So inside of each cell, there are long and complex molecules called chromosomes. Chromosomes is kind of like an instruction manual, and it's made out of DNA. They tell us everything we need to know to create a living cell. Part of this information can give us, for example, our hair color or eye color, and these sets of instructions are called genes. The genes, these sets of instructions are made up of small little molecules. And it requires a very specific sequence in order to give the proper information. And the proper order of this information is needed, or else it wouldn't provide the correct instructions. Every living thing, every cell in our body has 2 copies of each gene, one set passed on from your mother and the other one from your father. There can be multiple types of each gene, and they give different instructions, and one version might cause a person to be very tall, or one version might tell the person to be very short. These different versions of genes are called alleles. And since living things all have 2 copies of each gene, it can have 2 different alleles at the same time. So which ones get expressed? Which ones do you see? Well, the ones that get expressed are called the dominant gene, and whereas the one that doesn't express itself is called a recessive gene. Most of the characteristics in your body are a result of multiple genes. It's usually more than just one gene that would tell us a certain characteristic about you. And so far, the Human Genome Project has identified between 20 to 25,000 genes, and we don't quite yet know how all of them work and function together. So how does a defective gene give rise to a disease? Well, a gene mutation is a permanent change in the DNA sequence that make up the gene. Gene mutations occur in 2 ways. They can be inherited from a parent or acquired during a person's lifetime. Mutations that are passed from parent to child are called hereditary mutations. To function properly, genes depend on thousands of proteins to do their jobs, or cells depend on thousands of proteins to do their jobs at the right place at the right time. And if gene mutations prevent one or more of these proteins from working properly, then we get something called a genetic disorder. Usually these proteins either malfunction or they're missing completely. So which kind of leads me back to the patterns of inheritance. I mentioned recessive and dominance, but what does that mean? So genetic disorders can be thought of as autosomal dominant, meaning that just one gene, one copy of the gene is sufficient to cause the disorder. And autosomal dominant disorders tend to run in every generation. One example can be found in Retinitis pigmentosa. There's over 60 genes in RP, and approximately 20 of them are inherited in a dominant fashion, meaning that for that particular gene, when there's 2 copies of it in the patient, if one copy is affected, is sufficient to cause the genetic defect, and even though the other copy may be completely normal. RP is a result of damage to photoreceptors, particularly the rods, which are needed for peripheral and night vision. It occurs in approximately 1 in 3,500 people in Canada, so that's approximately 10,000 people in this country. That's quite a few patients. Patients would experience night vision problems at first, and then they would start getting progressive loss of their peripheral vision, followed by their central vision. This disease is very variable, and it may be diagnosed as early as childhood, and may even go undiagnosed until much later in adulthood. An example would be the patient that I described earlier. She's only 29 years old, and it has affected her deeply. Whereas my mother-in-law, she was diagnosed when she was 60. The next pattern of inheritance is autosomal recessive. And most retina eye conditions are passed on in an autosomal recessive manner. And it requires 2 mutated copies of the gene in each cell to cause that disorder. An affected person usually has an unaffected parent who carry a single copy of the gene that is passed on, and the other parent is also a carrier. They have also one abnormal copy of the gene that is passed on. So the child that has two affected copies of the gene now would express that condition. And autosomal recessive disorders are typically not seen in every generation, but usually skips between generations before we see it. An example would be Leber congenital amaurosis. These patients, when I look into their eyes, they have a retina that looks like retinitis pigmentosa, but it's not quite the same. And there's at least 14 genes that we know of that causes this condition, but these patients are usually affected in infancy. And the genes are involved in the development of the retina and also the signal transmission between the retina and the brain. And when there's a defective transmission, even if the retina is receiving light and receiving a signal, that signal unfortunately doesn't get to the brain and the infant doesn't see. In terms of retinitis pigmentosa, it's known that 35 of the 60 genes are known to be passed on in an autosomal recessive manner. Another autosomal recessive disease is Stargardt's disease. This affects about 1 in 10,000 people. And these patients get an accumulation of a material called lipofusin behind the retina in a layer called the retinal pigment epithelium. This layer is used to nourish the retina. It transports oxygen, nutrients, and supports the retina. This lipofusin is a material that is a result of a waste product of normal metabolism. We all produce it every day, but we have the ability to clear some of it, although we do accumulate slowly over time with age. And when we look at patients actually with macular degeneration, they have an accumulation of this lipofusin as well. So this accumulation of material causes photoreceptors, particularly the cones, which gives you sharp central vision as well as color vision, to slowly degenerate. Another pattern of inheritance is X-linked recessive diseases. X-linked recessive disorders are caused by mutations on the X chromosome. For the men, we have one X chromosome and one Y chromosome, and in women, we have two X chromosomes. So usually these conditions occur in males because there's only one X chromosome, whereas women are likely to be carriers of the disease because they have two X chromosomes. One may be affected, but the other one is normal, so usually they're not affected by the condition. A good example of a retinal degeneration is choroideremia. Choroideremia affects 1 in 50,000 patients, and they initially get impaired night vision, followed by progressive loss of their peripheral vision. Choroideremia, the genetic defect affects a escort protein in the cell. So imagine inside of the living cell, this protein helps transport and move molecules between different parts of the cell so that it can function properly. And so when it's missing this escort protein, the cells don't function properly and they die prematurely. Inside of the eye, in the back of the retina, there's a bed of blood vessels that support the retina, and this is called a choroid. And the choroid degenerates in these patients. And so with the loss of this bed of blood vessels, the overlying retina slowly dies. And lastly, there's a mitochondrial disease or pattern of inheritance. This type of inheritance is a little bit different. It's also known as maternal inheritance, and this applies to mitochondrial DNA. The mitochondria is a structure that's inside of every cell. It's it's there as a energy factory. It produces energy for the cells. And it contains a very small amount of DNA that's different than the ones that are found in your normal chromosomes. And because the mitochondria is passed on from the maternal egg, it's not in the sperm, in the eggs, it can only be passed on by the mother. And disorders from mutations of this small little segment of DNA can occur in every generation of a family, but it can't be passed on by men who have the condition. An example of that is Leber's hereditary optic neuropathy. This occurs in about 1 in 30,000 people, and they experience very sudden decreased vision in their teenage years or in their early 20s. The mutation of the mitochondrial DNA causes the cells in the optic nerve to die, so that even though the retina is functioning, again, no transmission of the signal is able to reach the brain. So that's just a very brief overview of some of the very common genetic conditions. But as you can see, it's very important to understand and to move forward with genetic eye diseases. There's still a lot that we don't know in terms of how certain mutations cause disease, and further research needs to be done. And that's where participation of all of you comes into play, because we want to find out where the defective gene is and what causes a mutation and how that mutation affects or causes the disorder or condition. So overall, in terms of retinal degenerative disease, there's over 200 different genes, and 2 patients who seem to have a very similar eye condition may have different mutations. And the location of these mutations can be identified using very sophisticated genetic testing and techniques. As you remember, each gene is made up of DNA and made up of hundreds of subunits, and those subunits are called base pairs. So mutations can occur anywhere along a string of these base pairs, so that even when 2 people have the vision loss due to an error in the same gene, they may have completely unique mutations. So why do we do this genetic testing? Well, first of all, genetic testing or molecular genetic testing or genotyping is usually done as part of genetic counseling. It involves taking a sample of your DNA from a blood sample and examining it in the laboratory. Genetic testing can give you and your physician more information about your condition, especially if the mutations are known. Some of the very well-documented mutations, we know in terms of the age range it starts affecting you, the progression of the disease, and how advanced the disease can get. But of course, testing doesn't always give you a clear result. There's still many mutations that we don't know about, and there's new mutations being discovered every day. So successful genetic testing can add to the information that people receive with genetic counseling. And part of the reason why we should do genetic testing is that, one, it confirms your diagnosis. It can tell you more about your prognosis. It can also help inform you about family planning decisions. For example, many people with autosomal recessive conditions or such as retinitis pigmentosa, they don't realize that their chances of passing it on to their kids are very remote, especially if their partner has no family history of retinitis pigmentosa. And so we don't want them to worry unnecessarily. With genetic testing, if there's a result, then patients can be put in line for participation in any clinical trials that may come up. And it also allows them to join the Foundation Fighting Blindness Patient Registry. So how can I arrange to get genetic testing? Well, genetic testing first requires a genetic counselor that works with you to arrange for the testing. This may be difficult in certain parts of the country, especially in more remote areas. Here in Ottawa, it's done through the CHEO Hospital, through the Children's Hospital, and their medical genetics department have genetic counselors that will work with you and send your blood to a laboratory that will do the testing. And certainly if the diagnosis is unsure of, or if there's questions of the diagnosis, it's best that the patient gets referred to see a retina specialist where hopefully some testing can be done to confirm the diagnosis before a referral is made to see a genetic counselor. So what information is needed? Well, information such as your name, address, phone numbers, the diagnosis. Very important would be information about your other family members. A detailed family history is often very helpful to make a diagnosis, as well as other tests such as a fluorescein angiogram or an electroretinogram or visual fields. All those things are helpful in terms of making a diagnosis. How long will it take? Well, for some of the more common mutations, there are very readily genetic tests that are done on microchips that look at a panel of genes at the same time. And those can be done relatively quickly, but results can still take months. For more rare mutations or new mutations, those can often take years, as retesting may need to be done, or it may need to be confirmed with other patients that may have the same mutation. And certainly with the testing, a mutation may not be identified, and then those blood specimens are stored frozen for many, many years until more is known about that gene or that mutation. Will my province pay for the genetic testing? And so once the patient is properly referred and through genetic counseling, I believe that the cost is covered. There may be instances where it's not, depending on the mutation or the genes that are involved. And can I pay for this myself? Well, yes. Genetic testing is becoming more readily available, and prices have come down significantly over the past few years, and patients can often get tested for more of the common mutations between $300 and $500, but for more rare things, it can be several thousand. So again, back to the results of genetic testing. The likelihood of receiving an answer from genetic testing will vary depending on your condition, and some conditions are associated with a small number of genes, and the genes are well known. Those are often the easiest to find. For example, patients with X-linked retinal schisis or choroideremia, they're very likely to get a result. Next would be patients with Leber congenital amaurosis or Stargardt's disease. And the most challenging would be retinitis pigmentosa, given the significant number of genes that need to be tested and the significant number of mutations that are available to be confirmed. So retinitis pigmentosa is a little bit of a challenge. So again, gene therapy is starting, and gene therapy requires injection of a virus that inserts a correct portion of a DNA into the cells in the retina. And these new instructions help produce the proper proteins that are needed for the cell to function. And surgery usually involves something called a vitrectomy, removing the vitreous from inside of the eye. And then the viral vector is then injected underneath of the retina, and these viruses will infect the cells with the correct DNA, and hopefully they produce the protein, and thereby prolonging the life of the cell so that it doesn't regenerate. It doesn't, however, cause the degenerated cells or the dead cells to come back alive. We can't do that yet. And the current clinical trial from my mentor, Dr. Ian McDonald in Edmonton, on choroideremia is going to start soon. He did receive Health Canada approval, and he's just waiting for an assessment on the purity of those viral vectors before surgery begins. So we're hoping that maybe at the end of the year or early next year that will be the case. And if that phase 1 trial works very well, the trial may be expanded across Canada and also to include Ottawa. So I'll leave it there, and I'll stop for questions.
I have diabetic retinopathy. Does this procedure help that disease?
So diabetes is there is a genetic predisposition, and it is multifactorial as well. So we don't know at present if a specific gene can cause a certain severity of diabetic retinopathy. So no, that's not known at the moment. Okay.
I have been diagnosed with Stargardt's disease, and also I was told that the photoreceptors are, this word term it was used, metabolized, or they are dead. And my question is, up until the treatment for it is ready, I would also assume that the photoreceptors, the number of photoreceptors in the retina is limited. Now, since they are dead, is it possible that, or is there a possibility that the disease will expand to other regions of the retina, or it is limited to the photoreceptors area only?
So in Stargardt's, it's mainly the photoreceptors that are in your central vision. And because the central vision, there's a higher metabolism rate, and so that accumulation of lipofuscin or material that causes the degeneration is greatest in the central vision. Okay, so there is a small degeneration in the periphery, but mostly it's concentrated in the center.
From the research, how successful is genetic therapy, and with what conditions? Is it like with Leber's and?
So with Leber's, they've clinical trialed on RP65, and they've done it on a dozen or so patients. And those patients that had the injection of the virus, certainly in that area of the retina where it was exposed to the virus, it has done well in terms of improved function. But they did find that the rate of loss of those cells was still no different than the disease. So they're still learning about it.
What about for cone dystrophy, which is kind of similar to Stargardt's?
Sure. So I'm not aware of starting of trials in cone dystrophy yet. I think there is, but I don't know of the results at present.
Hi. As a patient diagnosed with retinitis pigmentosa, where both parents were found to be carriers, is it possible to have more than one genetic mutation?
Yes. So there are forms of RP that patients carry multiple mutations. So yes, that can occur, and we're starting to find out more and more of those. Because we've always been looking for just one gene, but yes, there's definitely patients with separate mutations in separate genes that's causing their condition.
What's the difference between rod-cone dystrophy and cone-rod? I have RP, which is rod-cone.
Sure. So,
and this lady here has cone-rod dystrophy. I just want to know the difference.
Sure. So I see it as a spectrum of diseases, and they're caused by different mutations in different genes. And so patients with rod cone, their rods are more affected compared to their cones, but their cones are still affected over time. Whereas cone rod, there's a problem with their cones first and more of the rods later.
In your opinion, can a specific gene mutation, for example, the RDH12 gene, cause a certain disease in one individual versus another individual? For example, LCA in one person and, say, RP in another person?
Sorry, I didn't quite get the question.
Can a specific gene mutation cause a certain disease in one individual and a different disease in a different individual?
Yeah, so I think it has to do with one in terms of, yes, the mutation can cause disease, but if it's more multifactorial, there may be other factors that contribute to how the patient presents. So yes, I would say so. So I know there's definitely mutations where patients, they can be affected much earlier in age compared to someone with the same mutation that's much later in age. And some of those factors are still unknown as to why that occurs. So that's why we continue to do the research.
And so I'd like to thank Dr. Lee for sharing his knowledge with us today, and thank all of you for asking your questions.